find out which cluster that GUmbie would fit into. I hypothesized that it would be in F1 because I can compare my gel results to a database of all of the gels that have been found for other clustered bacteriophage. So when looking on the database, I found the F1 cluster, and I compared GUmbie to all of those members. One of such members was PMC, a phage in that cluster, and I found that it was very similar to PMC as well as the rest of the F1 cluster. I also found that GUmbie was similar to the F2 cluster, but there's only one member in that cluster, so it's really hard to differentiate between. There's about 10 members in the F1 cluster, so it's easier to compare. But you can see with EcoR1 there's very few bands. With Hay3 there's the characteristic small bands at the bottom, and then Claw1 is cut all throughout. So just by looking at the database, I was able to fairly easily determine which cluster that GUmbie would belong to. But to verify, I performed a polymerase chain reaction, or PCR, which using primers, I can determine which cluster that GUmbie would belong to. As Sammy was saying, PCR involves using a forward and a reverse primer that will amplify DNA in between. And so what we did was we purchased these primers that are specific to each cluster. And since I hypothesized that GUmbie would belong to the F1 cluster, then my results should show a band of amplified DNA using the F1 forward and reverse primers only. And it shouldn't show any bands with any of the other primers. These are my results from the PCR reaction. On the very left, again, we have a ladder with the various sizes of bands shown in base pairs. And then on the very right, we have a D29 control. D29 is a phage that is a member of the A cluster. And so the way that this is a control is that we know it's supposed to cut with A. And so we use the A1 forward and reverse primers just to show that I ran the PCR successfully. And as you can see, in the F1 lane, I successfully had a band of amplified DNA, so I was correct in my guess that it belonged to the F1 cluster. But there was a bit of a problem, because I also had amplification down here in the D lane, which I wasn't expecting. But you can notice that it's much farther below the last line on the ladder, and so I'm guessing that it's from primer dimers, which results when DNA isn't amplified, but actually what happens is the primers amplify each other. And then there's also another band over here, you can see it's kind of faint. So primer dimers happen in D amplify. And then down here at the bottom, you can notice some more blurry bands, and those are just the primers that are running through the gel. So really, looking at this, the only DNA that was actually amplified on the PCR results is from the F1 cluster, which verified my hypothesis. So the grand finale. After all of this was done, all of our phage were compared in the course, and based on the clear cuts in my gel, GUmbie was chosen to be sent off to have its entire genome sequenced, which I know doesn't seem that exciting, but to put it into context, there's only been about 1,000 phages that have been found, and out of that 1,000, only about 200 have had their entire genome sequenced, so including GUmbie. So it was really a big deal, and the reason that this doesn't usually happen is because it is very costly to have it done. But what's great about the phage program is that they sponsor to have one of our bacteriophage sequenced. So we can gain some more information about GUmbie and compare the genes to other bacteriophage. And one way that we can do this is by using the program called Famerator, and that's just a that's a bacteriophage comparison program where you can load the entire genome that we got that was sequenced and compare it to any of the other ones that have already been discovered. So just to show, this is what the Famerator program does. Each of the boxes that are shown are separate genes, and at the very top I have GUmbie, and this is really just the first portion of the genome. And what I did was I compared it to other members of the F1 cluster to see how related they were. And so there's Chi8, Pac40, Tweety, and Fulu that are all members. And if you see the purple highlighted regions, those are regions that are exactly identical to each other. So the genes are very similar. And then there's these blue bars and cluster because there's so much purple regions and very few white regions. So this is what it means to be in a family or a cluster. In conclusion, I hope that you can see from the beginning when I got my restriction enzyme digest results, I had a fairly good idea of what cluster that my phage belonged to, which was F1. And when I got GUmbie sequence, they also verified that it was NF1, so that's further evidence. And then they also verified that GUmbie is indeed a unique bacteriophage. It's never been discovered before. So it's pretty exciting, and this is just another representation of GUmbie on here. Are there any questions?
Let's take their, let's thank you.
Could you go to your PCR slide? Is there a way you can know that the F1 bend isn't also primer dimers? Like, is there any differentiating characteristics there?
To be honest, the way that we knew was because we had GUmbie sequence before we did the PCR reaction. So that's the only reason that I absolutely knew that it was. Otherwise, I would suspect that it was primer dimers just because it falls below the ladder. But that's the only reason that we knew.
I was wondering how you collected the bacteriophage.
Oh, okay.
Because I just know that you guys got them from around Gonzaga, but like, what's the specific method that you guys are using?
So basically we take soil about 5 inches deep that is moist because bacteriophage live in moist environments. They also live in the ocean or any bodies of water. And then we take the soil and we filter it, and we get a substrate that contains phage DNA, and then we go through a bunch of different purification processes to filter out all of the junk that's in the soil and just to isolate the DNA. And then we can purify the DNA and amplify it to get large quantities of it.
Can you go to your restriction enzyme, Jill? What was the reason for using Echo R1 twice?
I believe this one was a control that didn't actually have any enzyme in it. It was just labeled Eco R1. It had DNA in it, so it wasn't supposed to cut at all. So really, this lane just has DNA,